Namaste. My name is JVN Manisha Singh, Assistant Professor in Department of Food and Biotechnology under Faculty of Agriculture and Veterinary Sciences in Jyoti Vidyapeeth Women's University, Jaipur. Today I am teaching you food safety and quality control for class DET FBT 6th semester. In today's session, we will discuss all about the simple and quick method of food adulteration detection. And in previous session, we will discuss all about the introduction of food adulteration. So let's begin with the topic. The adulteration is a legal term, meaning that a food product fails to meet the legal standards. One form of adulteration is an additional of another substance to a food item in order to increase the quantity of food item uh, in raw form or in the prepared form, which may result in the loss of actual quality of the food item. Okay, food adulteration is defined as the practice of adulterating food or contamination of the food materials by adding few substances which are collectively called as the adulterants. Adulterants are the substances or poor quality products added to the food items for economic and technical benefits, and additional of these adulterants reduces the value of nutrients in food and also contaminates the food which is not fit for the human consumption. These adulterants can be available in all food products which we consume daily, including the dairy products, cereals, pulses, grains, meat, vegetables, fruits, oils, beverages, etc. Okay, so there are the tests for some adulterants in the food, like in milk and milk products. You can do the detection of water in milk, detergent in milk, and starch in milk and milk products like khoya, paneer, and chena. You can detect the starch in these food products, in the dairy products, and then detection of the mashed potatoes and the sweet potatoes in the milk products. Next, we have the oils and the fats and the detection of other oils in the coconut oil. Then sugars and confectionery, that is the detection of the sugar solution in honey and the detection of chalk powder in sugar, or you can say pithy sugar. Okay. Then food grains and its products like detection of extraneous matter, dust, pebbles, stone, etc. Detection of the ergot, detection of dhatura in the food products and the food grain, and detection of the excess bran in the wheat flour, detection of Kasri dal in the whole dal, or you can say split dal, and detection of the added color in the food grains, detection of iron fillings in Atta, Maida, Suji, and detection of turmeric in the sela rice, and detection of the Rhodamine B in the Ragi. And for the spices and the condiments, you can do the detection of foreign resin in the Asopotator, or also known as hing, detection of soapstone or other earthy matter, detection of papaya seeds in the black pepper, detection of artificial or water soluble synthetic seeds. Okay. Then we have the detection of cassia bark in the cinnamon, detection of grass seeds colored with charcoal, detection of Argimon seeds in the mustard seeds, and detection of lead chromate in the turmeric whole, detection of artificial color in the turmeric powder, and detection of the sawdust and the powdered bran in the powder, and the detection of extraneous matter in the spices and the condiments, and detection of the fennel seeds in the cumin seeds. And miscellaneous, there is a detection of malachite green in the green chili and the green vegetables. Malachite green is basically a dye, and detection of artificial color on the green peas, detection of the colored dye tendrils of the maize, and the detection of the white powdered in iodized salt, and the detection differentiation of common salt and the iodized salt, detection of clay in the coffee powder, detection of the color in the supari pan masala, detection of the exhausted tea in the tea leaves, and detection of iron fillings in the tea leaves, detection of chakunda beans in the pulses, and detection of rhodamine B in the sweet potato, and the detection of wax polishing on the apple, and detection of chicory powder in the coffee powder. So these all are the adulterants used in these respective food products. Okay, so there are the tests according to the FSSAI to determine whether the food product is adulterated or not. For the milk and milk products, we will do for the detection of water in the milk. So testing method is basically nothing. Put a drop on the polished slanting surface Pour milk on either stays or the flow slowly, leaving a white trail behind. Adulterated milk with water will flow immediately without leaving a mark, but the pure milk will trails with the uh, white trail, leaving the white trail behind the over the uh, polished slanting surface. Okay. Next, we have the detection of the detergent in the milk. So testing method is basically take 5 to 10 ml of sample with an equal amount of water. Shake the content thoroughly in the test tube. And if milk is adulterated with the detergent, then it forms a dense lather. And pure milk will form very thin layer due to agitation. Okay. Then we have test for the detection of starch in milk and milk products like khoa, chena, paneer. So testing method is boil 2 to 3 ml of sample with 5 ml of water, cool and add 2 to 3 drops of tincture of iodine, and formation of blue color indicates the presence of the starch. Okay, it is basically a starch reaction which we will do perform in our 6th class as well with the rice. Okay, so in the case of milk, addition of water and boiling is not required. Then we have the detection of mashed potatoes, sweet potatoes, and other starches in the ghee or the butter. So for that, you have to take half teaspoon of ghee or butter in a transparent bowl. Add 2 to 3 drops of tincture of iodine, and formation of blue color indicates that the uh, presence of mashed potatoes and sweet potatoes and other starches. Okay, because sweet potatoes and mashed potatoes are rich source of the starch, and with the in the presence of tincture of iodine, it will gives a blue black color. Okay, if it is pure, then it will not give any color. Okay, so if the butter and ghee is pure, then it will not leave any color. Okay, and if it is adulterated with these, then with any of these starches, then it will indicate the blue color, blue black color. Okay, next we have the oils and the fat. So detection of the other oils in the coconut oil. So take the coconut oil in a transparent glass. Place the glass in the refrigerator for 30 minutes and do not keep in the freezer. After refrigeration, coconut oil basically solidifies at the cold temperature, and if coconut oil is adulterated with any other oil, it will remain a separate layer. So it is a method of detection of other oils in the coconut oil. Next, we have the detection of sugar solution in the honey. So take a transparent glass of water and add a drop of honey to the glass. Pure honey will not disperse in water, and if the drop of honey disperses in water, it indicates the presence of the added sugar, because sugar dissolves in the water, but honey will not disperse or dissolve in the pure water. And next method is take a cotton wick dipped in a pure honey and light with the matchstick. Pure honey will burn, and if adulterated, the presence of the water will not allow the honey to burn. If it does, then it will produce a cracking sound. Okay. Next, we have the detection of chalk powder in the sugar or jaggery. So we require a transparent glass, and uh, basically we will transparent glass of water and dissolve the 10 grams of the sample in the water. Basically, it may be a sugar, jaggery, and the peaty sugar. And it when it is mixed with the chalk, the adulterant will be settled down. Okay, because the chalk powder is not dissolved in the water. It will basically separate the layer and it will settle down at the bottom of the glass. Next, we have detection of extraneous matter, dust, pebble, stone, straw weed, weed seeds, damaged grain, withered grain, insects, rodent hair, and excreta in the food grains. So take small quantity of the sample in a glass plate, examine the impurities visually, and pure food grains will not have any such impurities, and impurities are observed visually in the adulterated food grains. Okay. Then detection of the ergot. Ergot is nothing but a fungus containing the poisonous substances in the food grain. So for that, you have to put some grains in the transparent glass container containing the 20% of the salt solution and 20 grams of table salt. It may be iodized salt or a table salt in 100 ml of water. Then you will prepare a 20% of the salt solution. So put the grains into them, and ergot floats over the surface while sound grains settle down. So purple, black, longer sized grains show the presence of the ergot. Next, we have the food grains and its products. So detection of Dhatura in the food grains testing method is the take small quantity of the food grain in a glass plate, examine the impurities visually. Dhatura seeds are basically flat with the edges and blackish brown in color and can be separated out by the close examination. Next is detection of excess bran in the wheat flour. So take a transparent glass of water, sprinkle a spoon of water on the surface of the water, spoon of the wheat flour over the surface of the water. Pure wheat flour will not show excess bran on the water surface, and adulterated wheat flour shows excess bran floating on the water surface. Next, we have the detection of the Kasri dal in whole dal or split dal. Take small quantity of dal or split dal in a glass plate and examine the impurities visually. Pure dal will not have such any impurities. Kesari dal has the aged type appearance and showing a slant on one side and square in appearance, can be separated out by close examination. Okay. Next, we have the detection of added color in the food grain. So take a transparent glass of water, add 2 teaspoons of food grains and mix thoroughly. Pure food grains will not leave any color and adulterated food grains leaves color immediately in the water. Next, we have detection of iron fillings in atta, maida or suji. So it's nothing but take a small quantity of sample in a glass plate, move the magnet over the flour. Pure flour will not show any iron filling on the magnet. And if flour is adulterated, then the iron filling will be seen on the magnet. It's nothing. The principle is basically a magnet attracts the iron. Okay, so that's the principle. And detection of the turmeric in a sela rice. So take a teaspoon of rice in a glass plate, sprinkle a small amount of soaked lime, also commonly known as tuna, which is used in a pan or on the rice grains. Pure grains will not form red color with the soaked lime, and adulterated grains will form red color with it. Next, we have the detection of rhodamine B in ragi. So take cotton ball soaked in a water or vegetable oil. Okay, rub the outer surface of the ragi. If cotton absorbs the color, then it simply indicates the adulteration of rhodamine B dye for the coloring the outer surface of the ragi. Next is the spices and condiments. So that the detection of foreign resin in Asopotator is basically done by burning the small quantity of Asopotator in a stainless steel spoon. Pure Asopotator will burn like a camphor, but adulterated Asopotator will not burn like a bright flame like camphor. Okay. Next, we have the detection of the soapstone or other earthy matter in Asopotator. So testing method is shake little portion of the sample with water and allow to settle. Pure Asopotator will not leave any soapstone or other earthy matter at the bottom. If Asopotator is adulterated, soapstone or other earthy matter will settle down at the bottom. Okay, next we have detection of the papaya seeds in the black pepper. Okay, so testing method is add some amount of black pepper to a glass of water. Pure black pepper settles at the bottom because it is heavy, and in the adulterated black pepper, papaya seeds float on the surface of the water. Next, we have the detection of artificial water soluble synthetic colors in chili powder. So sprinkle chili powder on the surface of the water taken in a glass tumbler. The artificial colorants will immediately start descending in the color streaks. Okay. Next is the detection of cassia bark in a cinnamon, which is basically a spices, and take small quantity of cinnamon in a glass plate if adulterated. On close visual experience, cassia bark that comprises of several rails in between the rough outer and innermost smooth layers can be differentiated from the cinnamon. Cinnamon barks are very thin and that can be rolled around a pencil or a pen, and it has a distinct smell. Okay, then we have the detection of grass seeds colored with the charcoal dust in cumin seeds. So rub the small amount of cumin seeds on the palms. If palms turn black, adulteration is indicated. Next step, detection of Argimon seeds in the mustard seeds. So the testing method is take small quantity of the mustard seeds in a glass plate, examine visually for the Argimon seeds. Then mustard seeds have a smooth surface and when pressed inside it is yellow in color. And Argimon seeds have grainy rough surface and are black in color. When pressed inside it is white in color. Okay. Next we have detection of lead chromate in turmeric whole. So add a small quantity of turmeric whole in a transparent glass of water. Pure turmeric will not leave any color, and adulterated turmeric appears to be bright in color and leaves color immediately in water. Okay, so from these questions, various questions may be asked, like explain the detection method, detection of papaya seeds in the black pepper, and explain the detection methods used for the milk and milk products and spices and condiments. Likewise, questions may be asked in your final exams, so prepare accordingly. Um I hope it is clear to all. Thank you so much for watching this session.